Hello everybody, I'm Dr. Indira from the Department of Life Sciences, Christu Jayanti University. In my lecture today, I shall be talking about the various stages of micropropagation in plant tissue culture. Let us understand what is micropropagation. In plant tissue culture, otherwise referred to as micropropagation, is a method of artificially producing plants in a nutrient medium where we produce mass number of plants by asexual method of reproduction. The stages of micropropagation involves several methods like selection of an explant, culture initiation and establishment, shoot multiplication, rooting of the shoots, and transfer of the plantlets in the greenhouse environment. Let us look at these stages one by one. In the initial stages of micropropagation, the stock plants that serve as explants are grown in a suitable condition in the greenhouse. In the next stage, the explants are subjected to a few steps before we inoculate them into the nutrient media. First, we have to isolate the explants, which could be a leaf, node, internode. The choice depends upon the type of culture that we intend to. The explant is then further subjected to surface sterilization, where they are initially washed with water, tween 20 as a detergent, and then they are further taken into the laminar airflow, where they are treated with 70% alcohol and other surface sterilants like mercury chloride or sodium hypochlorite. The explants are repeatedly washed with sterile distilled water, and then they are inoculated into an appropriate culture medium containing the required hormone concentrations. Morashiki and Scoops media is the most ideal plant tissue culture media used for micropropagation. Depending upon the explant, the proportion of auxins and cytokinins may be altered. The nature of the explant may vary. It could be a leaf, it could be a node, internode, and accordingly, the choice of the hormones are used. After sufficient multiple shoots are produced in the nutrient media, which may be through direct organogenesis or indirect organogenesis, which may take about 4 to 8 weeks. These shoots are then carried over to the next stage for further development. After sufficient multiple shoots are produced in the culture medium, the shoots are now transferred to a medium for the development of roots. Individual shoots are now transferred into the hardening mixtures for root development, or they are transferred to a nutrient medium in the laboratory. Now, the hardening mixtures may comprise of coco peat, soilrite, perlite, sand, either individually or they may be chosen in combinations. The following procedures are to be maintained while the rooting process is done. Every shoot should be able to root individually. The hardening of the shoots is basically done to increase their resistance, allowing it to become more stronger, allowing the plants to be capable of being autotrophic rather than heterotrophic while they were in the culture medium. They should be able to fulfill the requirements of breaking dormancy. In the next stage, after the plants have successfully rooted in the hardening mixtures, they are now transferred to the soil, which is maintained in the greenhouse at ideal conditions of temperature, humidity, and light. Let us now look at the advantages of micropropagation. Micropropagation offers an alternative method of vegetative propagation with enhanced multiplication rate. Large quantities of plants can be produced within a very short time from a single plant tissue. The shoot multiplication has a very short cycle, and each cycle results in an exponential increase in the number of shoots. The small size propagules can be stored and transported easily. The germplasm stocks can be maintained for several years using this technique. Micropropagation allows the production and maintenance of disease-free plants. Millions of plants can be maintained in the culture bottles or tubes. Genetic uniformity of the propagules can be maintained through this cultures or technique. Of course, it is a cost-effective process, but new varieties and species can be propagated. It requires less space and human resources. This method is independent of season and can be carried out anytime. This micropropagation technique assists in regenerating genetically modified cells after protoplast fusion, often produces healthier plants, leading to quicker growth compared to those plants which are produced by conventional methods. Apart from several advantages of micropropagation, it also has some limitations. The plants may sometimes not be able to adjust itself to the environmental conditions and thus may not be autotrophic. This technique may not be implemented in all the crops. The plants may find a problem acclimatizing to the natural environment. I hope this information was useful. Thank you for watching.